Hello, I'm here for the second member spotlight for the ISOP newsletter. And today I am with Sina Adl, and we're going to talk about his current role as president of ISOP and also some information on his protostology textbook. So I like to start these off pretty much with the question, what led you to study protostology?
I guess it was second year cell biology when I realized that if you really wanted to get a handle on cell biology, all of the diversity was in the protist for eukaryotes.
Yeah.
And without getting a handle on protistology, you wouldn't really get a good scope of all of the diversity there was across eukaryotes. So kind of went into protist cell biology and eukaryotic cell biology from there.
Cool. What was there like a specific protist that you started off
Not
with,
really.
or?
Just decided to second year cell biology to focus on protistology, and so I started looking for profs who were in the area, looking for summer students. So my undergrad summers were spent in protistology lab, doing various things, some sampling, some genetics, some cell biology, molecular biology.
Cool. So, yeah, your current role is president of ISOP.
Yeah.
So, did you want to talk about some of your priorities and goals of this presidency year?
Sure. I think a lot of the tasks kind of get handed over from one president to the next. So there are a variety of things we're working on last year, primarily renewing the journal contract with Wiley. And then they were introducing a lot of corrections, revisions to the bylaws. So those went through last summer. And so now we're trying to work with this new committee structure and executive structure. So that requires some changes in what committees do, and we're fine tuning that a little bit.
Nice.
And we have ISOP is a bit of an umbrella society for a whole variety of other societies like ISEP and some national organizations, testing to maybe people. But we are so one of the things we do is make some money available to these organizations every year to support students or conferences or some of their events. But we have to renegotiate uh, what their benefits are, how they pay, what they pay, and what ISOP's responsibilities towards all these other national organizations. So those discussions started about a year ago, and they're still ongoing. But I think during February, March, we'll finalize it and have a new fee structure in June for everybody. So this is making it a lot. We've reduced the membership dues quite a lot, and we're going to, I hope, make it simpler for affiliate societies and how their interactions with ISOP are managed.
Yeah, yeah, like that makes like
Some of
so
the
much
biggest.
sense since
And then
there's
the
like,
society
yeah,
never hasn't had a strategic plan for a long time. So we're looking at uh, patterns in our membership numbers and revenues and working on a strategic plan for the first time in a long time.
nice. Yeah, like it makes a lot of sense since there are all these like other organizations that are hosting these protostology associated events. So it makes
Yeah,
sense to just kind of like connect them better, and ISOP's like the perfect like
yeah.
vehicle for that. Cool. Um, so how long have you been a member of ISOP, and has ISOP really like directed your, well, like helped out
Yeah.
your career at all, or?
So I was an undergraduate and I started going to ISOP in 1991. Um, I published my first paper in 91 when I was an undergrad. And so this kind of became my main society from there. In 94, the president at the time started strategic planning exercise, and he put me as the student rep on that committee. So I got involved with the executive 1994, and have been involved with society executive most of the time since. I took a break probably around 2013, 14, until a couple of years ago.
Yeah, and you've served as president like a few times.
One time before, I think around 2012.
Nice.
Yeah.
Do you notice like any like major changes with ISOP since you joined
Oh yeah,
a
quite
while
a lot.
ago?
So um, um, a few things changed. Some things. I think that was the beginning of our interactions with the affiliate societies and trying to make them more involved and more part of the whole team, I guess, of colleagues around the world. And then returning about 10 years later to see how that developed and how it needs to be adjusted again.
Yeah.
Because they've been involved with us for about 10 years, but things have changed and our relationship has matured a bit, but we need to have a different arrangement because of publishing has changed, access to journals and papers has changed. Things were harder to obtain online then, and it's much simpler now. If you compare 2005, 2015, 2025, are entirely different online worlds and
Yeah.
access. And since COVID, relationship between conferences and societies and membership has changed. So we have to adjust those again, and it's not always obvious or easy.
Yeah.
Yeah.
Cool. And kind of switching gears here, but I know you're working on or you've worked on a protostology textbook. And I think that's awesome for the field because that's really like people starting a course in protostology, there isn't really an option of a textbook to use in most cases.
Yeah.
So did you want to like talk about the development of that textbook?
I guess the hardest part, I've been meaning to write it for a long time, but the hardest part was making time for it because it's a massive undertaking and you don't have a book to base it on to
Mhm.
revise or update. Um, and what I noticed over time is fewer universities offering protistology courses. Microbiology became bacteriology. They may or may not be mycology courses, but the protists really completely fell out of university teaching because the classification changed a lot, the words changed a lot. Nobody was keeping track except for some specialists, and you lose the connection between the new words with the old words in the literature. And the grad students had a narrower and narrower perspective on protistology, where more and more grad students knew their group or a few families in their group,
Yeah.
and a very poor overview of what the other protists were, how they were related, what they did. And so eventually, during COVID, I had time, so I started writing the first chapters about what those groups were, and then I got a book contract and set up a sabbatical. So I had about 18 months with free time,
Yeah.
and continued with the book. And it was essentially a full-time project. What I wake up in the morning, start writing. When I'm too tired, go to bed and stop writing.
Yeah.
That took about 3 years of
Yeah.
maintaining that routine
Wow.
to get it done. So it ended up being quite a big book in the end. So
Yeah.
I don't know if you saw it, but it's quite big.
Yeah, but it's done, right? So
Yeah. So
Yeah,
it
and like what
was finished August and finished doing the corrections to the chapters and proofs in the fall, and they went to the printing press last week of November, and I received my copy mid-December.
awesome. Yeah, and like what
Yeah,
you're
that's
saying is
finished. It was exhausting.
yeah, yeah. Congrats. And it's like so apparent like what you were saying about like how protostology is taught and everything because when I did my PhD and undergrad at University of Miami, pretty much most of my microbiology experience in general was just like related to humans or like diseases of humans.
Yeah,
And the closest like parasitology, well, like the closest protos I got was parasitology. And it's just like you lose like so much from not seeing them from like an ecological and evolution perspective. It's just
yeah,
crazy. And then a lot of the biology textbooks, they're all based off the like protostology of like the like 60s and 70s and stuff, or like how we understood
yeah.
the evolutionary tree and stuff, but it's all new now. So that's really
I think
important. Are there any like
I'll
yeah,
teach it, and I think it'll help grad student get a handle of the past literature,
yeah,
because I've tried to use the same words as much as possible consistently throughout. If you go to some of the algae fungal literature, every time you switch group, they tend to switch the vocabulary, and so you have to, sometimes you don't realize that a cyst here is a spore over there,
yeah.
and trying to streamline that vocabulary gets rid of a lot of the confusion.
Yeah.
I think makes it simpler to learn and understand and then move forward. And then when you read the old literature, you can say, oh, yeah, well, we call that a cyst these days.
Yeah.
Yeah.
And then like trying to distill like all of protostology into a textbook, were there like some central themes you focused on, or like
Um,
what's the main thing you want like someone to like take away like
yeah. So
from
I think
the
for
course?
section 2, I start with origin of eukaryotes. And then I look at diversification of the protists once there was a eukaryote.
Yeah.
Um, so that really takes a more mechanical evolutionary perspective on what happened next. So there's a chapter on getting the first plastid. There's a chapter on getting secondary, tertiary plastids. There's a chapter on losing organelles and losing mitochondrial function and adaptation to anaerobic life or parasitic life. There's a chapter called touching, feeling, sensing, and that deals with chemotaxis and cell membrane potential difference things and behavior, but it also has all of the parasite life cycles, because the parasite has to recognize a host. It has to recognize host tissues to get into the correct host tissue and then come out of it and go to another host tissue.
Yeah.
And all of that is about feeling your way in your habitat, in your environment where you are. So it's all about cell membranes, touching cell membranes and feeling it and sensing where they are.
That's awesome. Yeah, like I'm excited to like eventually like see a copy of it. Like I don't know if like what the distribution plan is for the textbook, if there's like
Well, I think a lot of university libraries already have it.
oh cool.
It exists online as a textbook. So most, especially larger universities, I think most universities have it online if you go through the catalog. Um, and a lot of universities have already ordered their copy, so they probably have it or in process. Smaller universities might take a bit longer. Yeah,
Cool. Yeah, so I'll check it out here at UBC. So
yeah. Or ask them to get it if they don't have it.
yeah, exactly. Yeah, since I think like yeah, it's one of the few like universities that also like teaches protostology as a class
Right.
too. So it'd be
Yeah.
definitely useful.
Yeah, we're lucky in Canada. There's a group in at Dalhousie. There's a group at UBC. Those are two areas of strengths in protistology. In the Czech Republic,
Yeah.
there's a lot of strength there, especially in České Budějovice, huge groups there, and in Prague. In France, there are a couple of marine stations that have a lot of protistologists. So
Yeah.
a small group in the UK.
Cool. So like as a field, like protostology, what kind of like challenges do you see us facing in the future that we should like adapt for or be prepared
Well, I think
for?
this is a very exciting time. The late 90s, it was a period when protostology was not really being funded anymore. Everything was switching to cancer research,
Mm,
more important parasites. And then as we progressed through the 2000s, and things stabilized with regards to understanding phylogenetics and what the protist cell biology was. I think of the classification a bit like chemists think of the periodic table. Once you organize things the way they're supposed to be organized, you see the patterns more easily.
yeah.
Gain of this and then secondary loss of that. And so those patterns become more obvious when you're tracking cell biology. Once you have a good classification or a solid phylogenetic tree. Um, so we have a combination now where we have a stable understanding of what the phylogenies are, what the classification is. But we also have a lot of new techniques that make single cell biology, genomics possible. And those were developed for cancer research and other purposes. But I think protistology labs have really adopted those well.
Yeah.
If you look at Inyaki's lab, for example, been doing it for a while now. So we have the things that were a handicap. For example, when I was doing my postdoc, we were looking for these rare cytoskeleton fine elements that were involved in cancers in humans, and they were abundant in some protists. And we were trying to get at them with the cell biology, molecular biology at the time, but we couldn't. The techniques were not there.
Yeah.
Um, but if I wanted to do that project now, there are techniques that you would go to that work, so you wouldn't have that handicap anymore. And so I think a lot of the genetics, molecular biology that were a problem 20 years ago, 25 years ago, now are completely accessible.
Yeah.
You can make most things into a model organism just by spending a few months at it.
Yeah.
Whereas before you had to spend, years and years and years getting homozygous lines and manipulating the genetics, figuring out how to put DNA in and out of it, and sometimes you couldn't. Now everything is much, much simpler. It's a matter of getting one clever grad student to fix it.
Yeah.
And so I think it's a very exciting time because a lot of things are more difficult to do with parasites or in humans or in vivo. We can
Mhm.
now do with single cell genomics and molecular biology techniques. So I think it's going to be about 10, 15 years of very exciting time for us.
Yeah. Yeah, that's awesome. And a good point with like a lot of the like cancer research and stuff and just adapting
Yeah,
protocols for like human models and other things towards protists. And it just takes a little bit of finagling and fixing, but the technique
yeah,
works usually. So
yeah.
awesome.
And then we have, of course, a lot of, we're much better at digging through environmental DNA sequences. And so there are all these novel species out there and OTUs that you have, but you don't know what the bug is.
Yeah.
And
Yeah.
so those now with environmental sequences available, we can go specifically looking for some of these things that are missing that we need to know. And I think the Tara Ocean project with the marine samples really made it clear what you could do if you had a big enough sampling project. One person can sample once in their life and then spend 20 years playing with the data.
Yeah.
Um, so that those are, I think, again, exciting times if you're trying to see how the deep sea sediment works. Places that have been more inaccessible, a lot of terrestrial places are unsampled completely.
Yeah.
and we have way more diversity in the soil than we have in water. It's so much richer with nutrients.
Yeah. Yeah, true. And some of those like binning techniques that are used for like prokaryotic genomes, they're being adapted now for eukaryotes, and that opens
Yeah,
up like the whole realm of other samples. So
yeah,
that'd be cool.
yeah. I think hopefully the book has a lot of ideas for thesis projects where I'll say, well, I don't think we really understand this well.
Yeah,
And I'll say, well, there are 12 theories explaining this, but none of them work. None of them, they're all obviously false. So we need someone to come up with something else.
nice.
So I think there are, for people who go through it in areas in the chapters they're interested in, there's ideas for thesis.
That's awesome. Yeah, and like it's awesome that our field is like that too. There's so many open questions that any like excited student could just like hop on a project and
Yeah.
tackle that.
I think our biggest limitation is not interesting questions. It's a lack of funding. So I
Yeah,
think grad students will increasingly move towards countries where there is more funding.
yeah,
Some these are cycles every country goes through, with more and then less research funds, more restrictive, more permissive research funds. And so you see these shifts around the world where the grad students accumulate and do more work.
yeah. So, are there any specific mentors you've had along your career path that kind of like influenced
Yeah, I
your
think at
current job?
UBC, I was at UBC 15 years. And there were people there I spent a lot of time talking to, like Jim Berger, who was my supervisor all those years, and Max Taylor,
Mm,
but also in chemistry with Lionel Harrison, Beverly Green in botany. Um, I think in those days they were very influential. Mathematical biology, there was Leah Keshet that would always go and bug her about some models and mathematical formulations. And then others, I think over the years, Andre Adout and his group in France
yeah,
went there a couple of times. A lot of molecular biology techniques in Janine Besson's group in Paris, and then for soil ecology, like moving into the protists in the soil environment. I think Dave Coleman at UGA in the States, Athens, Georgia. Spent 2.5 years having lunch with him every day, learning more stuff.
yeah,
That was a huge influence. Mm.
yeah. There's like so much you can learn just from like informal di discussions over lunch, pretty much. That like,
Yep.
yeah, I feel like every lab just
Then,
kind of needs like an
of
informal
course,
time
like
to like
Cavalli
share
Smith,
ideas.
I met with him
Yeah,
at least 1, 2, 3 times every year and we always banged our heads over different things because I rarely agreed with him.
yeah,
Sometimes I did, but a lot of times I didn't.
yeah, and that's the like fun part of protostology, just those debates and stuff.
Yeah.
So, do you have any advice for early career researchers?
Well, I think right now, the comparative cell biology is for sure a place where there's a lot to contribute. Understanding evolution of metabolic pathways helps understand uh, niche specificity and nutrient uptake across the ecology.
Hmm.
So for ecologists, talking about metabolism sounds weird, but if you want to do microbial ecology, it's all about metabolism and what the species can do and cannot do,
Yeah.
and then from there how they relate. And then from a molecular biology perspective for disease, understanding disease and managing diseases, there's a lot that can be done now that would have been very difficult 10 years ago and
Yeah.
maybe you think of these experiments that you couldn't do 15 years ago, whereas now we can actually address these much more elegantly. So for understanding a lot of these eukaryotic diseases, and some of them are, like diarrheas and blood diseases.
Yeah.
All the intestinal diseases are important in everything that's agricultural livestock.
Hmm.
Those are going to see a lot of progress and finding ways that we can approach making vaccines. Now that we can make vaccines against some malaria, some trypanosomes,
Yeah.
we're going to be able to handle a lot of these apicomplexan parasites better. Um, so I think, there's a lot to do.
Cool. Are there any like projects you have planned or ones in the future that you're particularly excited about or
Next
that
three
you're
years,
willing to share?
I
Yeah.
actually have a couple more book contracts. So one of them I'm
Nice.
finishing 2025. The other one will be finished 26 or 27.
What are they about?
I have another sabbatical in 27. So between now and then, my focus really is on finishing these books.
Yeah. Are they like focused on specific themes or
Yeah. One of them is microbial ecology,
nice?
introduction to microbial ecology, which is going to be the last one to finish. And then next one will be finished this year is soil ecology, more specifically for the soil. Um, so those, I think they're going to take most of my time next 2, 3 years.
Yeah. Well, thank you
I
for
enjoy
your
doing
time.
this.
Yeah, yeah.
I find
Like
it less
thanks for
stressful
sharing.
and productive use of time. Um, I probably spent 3 years, 20 since COVID, so 2020, 21, 22. Spent a lot of time stepping down from committees and admin responsibilities. I had a lot of those,
Yeah.
um, and started focusing on getting some of these books done because I think I just find it a better use of my time.
Yeah, yeah, and it's very
More
like
rewarding.
necessary and yeah, and helpful for the future generation definitely. Like
Yeah, I find I'm a lot
finally,
more in control of my schedule and what I do every day compared to when I was doing a lot of administration where you have almost no control over your days.
yeah.
And then you on top. Um, so, and having a large research group takes pretty much all of your time. So can't really have a large research group at the same time as spending your time doing research. I think those some people might be able to do it, but I think my headspace just needs to really get isolated and buried in the book.
Yeah. Well, it's definitely like worth the endeavor and everything. So thank you. Well,
Yeah,
I
I'm
appreciate
curious to
your
see
time. Yeah.
people next summer at conferences and
Yes.
get into some discussions about all the things I said in the book that were wrong.
Yeah. So at ISOP in Korea this
Yep.
summer, so
Yeah,
that'd
I'll
be
be there
cool.
for that.
Awesome. So yeah, I think we went a little bit past our usual time for these, but I think we covered a lot of ground and I'm excited to get my hands on your textbook eventually. So yeah, and I appreciate your time.
Great. Yeah, let me know if you have more questions.
Yep.